Welcome back to our Intro to Flow Cytometry series. If you missed our last episode, I'll put a link up in the top corner so you can check it out. Today, we're going to be focusing on fluidics. You see what we did there? Let's dive in. The fluidic system of your flow cytometer brings your sample from your tube through the machine so it can be interrogated by the lasers. It may seem like a pretty simple concept, however, poorly functioning fluidics will always lead to bad data, so it's important to understand how they work and how to harness the power of your fluidics. The magic of the fluidic system happens at the flow cell. This is where your sample gets injected into the machine and meets the sheath. The sheath is flowing alongside the sample. Importantly, the two do not mix. And the sheath then is what brings your sample through the machine, narrowing the stream so that your cells are passing by the laser one cell at a time in the proper position so that the fluorochromes can be activated and resolved. It's kind of like focusing on a microscope. If your cells are outside of that boundary, you're going to lose some of the resolution of your signal. How this focusing is accomplished is through something known as hydrodynamic focusing, which works on the principles of laminar flow. So basically, your sheath is flowing at a faster rate than your sample and in a cone shape, which focuses your sample into a narrow stream. And this enclosed outer layer of sheath creates essentially a drag effect to bring your sample in its core stream through the machine. On every flow cytometer, you're going to have the capability of changing your speed. Now this will be potentially buttons that say low, medium, and high, or it could be a toggle switch inside the software to change your speed. Either way, the effect is the same. In order to get a faster run of your sample through the machine, you're pushing your sample through the machine at a higher pressure. So essentially, the different flow rates increase your sample pressure, which increases the width of your flow core where your cells are going through, that central stream. So essentially what you're doing is taking a nice narrow stream and making it larger and larger and larger as you increase your pressure. Now, this can have some very serious side effects for detecting your cells. First and foremost, you're going to increase the chance of coincidence, which is the chance that more than one cell passes through the laser at a time. And these events can then be detected as a singular event potentially, which can really affect your data. A higher flow rate also increases the likelihood that your cells will pass by the laser off beam or out of focus, out of the focal plane. And because of this, you can lose some of the resolution of your data. So if we look at a cross-section of a stream at low, medium, or high, you can see at low pressure we have a nice narrow core stream that only really one cell can pass through at a time. As we move to medium pressure, this center core gets larger and more cells fit inside it. And at high, this gets even larger. And you can see many cells can pass through at any given time. If we then switch to look at the plane of focus, you'll see that at low speed, all the cells are in that core central plane of focus. And as our speed increases, more cells are detected outside of this plane of focus, which will affect your resolution. So now that you understand the purpose of your fluidic system, here are some tips so that you can optimize your fluidics during your experimental runs. Tip number 1, start with a full sheath tank. Fluidics require consistent pressure. Starting with a full sheath tank will help you achieve this inside the machine. Tip number 2, keep your machine clean. Debris, dirt, cells build up inside the fluidics lines are fluidics' worst enemy. Meticulously clean your machine to remove these between each run. Tip number 3, run on low. I say this and I know most of you will not follow it. Low will give you your best run possible. However, it is the slowest. So if you find that your samples are running slowly, I recommend instead of turning up the speed, go and concentrate your samples. However, knowing that a lot of people will not always run on low, 3 with an asterisk, at least keep your speed consistent and run all your samples at the same speed so that all your samples are subject to the same loss of resolution by increasing the speed. Tip number 4, monitor fluorescence over time. This really will show you how your fluidics are functioning inside the machine. So if you look at this plot here, you'll see that normal fluidics, you'll have nice straight lines across. So you'll have your negative population and your positive population. They both move across time without jumping, shifting, or losing signal. When you have, say, a clumpy sample like this one, you'll notice that you have gaps in your run where your fluidics are running, stop, they jump a little, start again, jump a little, start again, and go. This indicates that you had poor fluidics during your run. So your fluidics will tell you all sorts of issues, like if you had a clumpy sample, if you clogged the machine, if you had issues with your laser delay. All of these things can be assessed by looking at fluorescence over time. And finally, and this rule stands for all of flow cytometry, tip number 5, perfect your sample prep. Your flow cytometry data is only going to be as good as the samples that you put into the machine. Or as we like to say, garbage in, garbage out. And with that, thanks for joining us in our second episode in our Intro to Flow Cytometry series focusing on fluidics. Make sure you tap the subscribe button and hit that little bell icon so you get notified when our next video drops. And with that, we'll see you in the next one.